Thank you for coming to my talk today. Actually, it's pronounced BIOCRATES. If you can think of Socrates, that can help you remember that. Anyway, today I'm representing BIOCRATES. We're considered to be a deep phenotyping company, and I'll explain a little bit more about that. We're focusing on metabolic phenotyping, which has broad applications for diagnosis, treatment prediction, and system diagnosis. We're located in the beautiful area of Innsbruck. The company started around 2000. It's basically an academic initiative, fully integrated mass spec platform for targeted metabolomics, biomarker. We have been involved with detection of biomarker. I left out Alzheimer's, which was just recently discovered here a couple weeks back now. And we established the first analytical service lab. And we launched our kit service around 2008, commercially available. What that means is that you can take a P180 kit, for example, and run it in your lab in-house. And the numbers here, we're a very small company. So we provide the kits and contract services. We have 14 or so assays. You can send samples to BIOCRATES, and we can provide a full service in terms of data presentation to data analysis and interpretation. So this is kind of the agenda I would like to touch on. I know I'm limited to time, so I have a lot to share with you today. So deep phenotyping, potential in diagnosis, prediction, system biology, specifically in these GWAS type studies. What we're understanding now is going full circle to biochemistry, system diagnostics, metabolites linked to pathophysiology and disease, and BIOCRATES metabolomics technology and products. I'll touch over those. So what we talk about deep phenotype, what we're trying to say here is that in a living system, they have a program, a genetic program. And then for us as humans to understand what that really means, the closest thing that we can get to that is kind of looking at the phenotype, diabetes, cancer, and often it's too late to kind of intervene. So what we're trying to do now is to say, let's look at these small molecules, and we have a closer handle on that being closer to the phenotype. So I'll give you some examples of applications in diagnostics and predictions. And so here's some data I wanted to present to you. It's actually not published, so I asked if we can present this. What I wanted to just show you here is that in this particular study, they were able to distinguish HIV patients from breast cancer patient compared to controls. And then in a caloric restriction and non-caloric restriction mouse, they were able to distinguish the metabolic profile here clearly. And then furthermore, you're able to discern the differences between the stages of breast cancer from stage 3 to stage 5. Here's another paper I'd like to talk about just briefly. Basically, it's diagnosis of breast cancer, and what they were able to do here was to identify a training set that will clearly distinguish between controls and the breast cancer patient. And then they did a validation set here between the controls and the patients. And with that, they were able to use logistic regression and devise a formulation with a sensitivity of 98.1% and specificity of 96%. Here's another paper on endometriosis, which is a very hard to get at type of diagnosis without doing actual visual inspection of the vaginal cavity. And typically it takes maybe 12 years or so before you get some kind of diagnosis from these patients. And you can think about that, it's 12 years of damage. So the idea is if we can predict or develop a way of actually understanding what's going on earlier on, and that's why they were using metabolomics in this stage. And what we are showing you here is in this RAC curve, using these metabolites, we're able to improve upon the RAC curve by 90% of sensitivity and 84.3% specificity. And this is the recent paper I alluded to earlier. It's the Alzheimer's paper. It came out maybe 2 weeks ago in Nature Medicine. And we understand some of the burdens of Alzheimer's. And so what's so important about this, it's one of the first efforts to identify biomarkers in blood. Typically, we have these subjective ways of determining the severity or progression of Alzheimer's. So it's a pretty big deal, and it's actually in line with going towards the NIAA requirements for determination what a biomarker is. Here's another paper I'd like to talk about just briefly. Again, it's schizophrenia. If you notice, these are some common things that I'll show you, these RSC curve, where it really kind of lends itself to more clinical applications that most of the clinicians are familiar with. And again, these are some difficult indications to understand. Typically, they have been subjective, and now we're looking at the small molecules to discern groups. And here what they were able to show was there were a set of it's dead. there were a set of metabolites at the bottom here that was able to then improve upon the prediction for cases and control here when you add that to the analysis. Again, so many papers have been come out of type 2 diabetes, and what I'm showing you here, again, using metabolites, we were able to then improve upon the predictiveness of the patients controlled against people who have diabetes. Here's a paper. It wasn't done with one of our kits, but what it's trying to do here, we have other methods, the assays. It's showing you that metabolites approach for predicting response to neoadjuvant chemotherapy for breast cancer. If you can understand, there are patients that don't respond to chemotherapy. And so if you can identify those group of individuals, you can then provide them with alternative methods to help them. And so what this paper is showing you here is that we're able to improve upon the predictiveness of those individuals. I think it was 80% of patients whose tumor did not respond to chemotherapy. Again, so what we're showing here, another breast cancer study, and they were using resveratrol, which is extracted from grapes, and it's an anti-cancer agent. And what we're showing here, the importance of genetic background. So we have a cell line, MCF here, and the other cell line here, and both of these are either responders or non-responders, have antiproliferative properties. And we can show you here that the two breast cell lines respond differently. They have a different profile based on their genetic background. What we're looking at, Vladimir spoke about study design earlier. Here's a study design. We're trying to understand mode of action. Typically, there's some time sequence involved with those. And so very important in terms of preclinical development. We want to understand if this is a drug that we want to take forward. So here's a study just showing you in terms of type 2 diabetes mouse. They were able to show that the amino acids in the plasma and in the controls and in the diabetes mouse model. And we are showing you here the changes in the metabolites that were normalized across the zero line here. And what this is showing you here from the same study is that the rosestilazone nor the drug improved upon hyperglycemia in the mouse models. However, the drug candidate, but not resveratrol, improved short-term metabolomic control to a level of healthy controls. So this is the dawning of system biology. The first paper came out about 2008. They combined GWAS with the targeted metabolomics approach. And you may be familiar with these heat maps. And what was uncovered, which they had subsequently followed up with a Nature paper, was to identify this this enzyme here, FADS1, and it was associated with this particular fatty acid here, PCC36-4. And to improve upon that association, what they looked at was looking at the ratios of these metabolites. So what this was actually doing here was acting as a surrogate for enzymatic activity by looking now quantifying the substrate and the product of that enzymatic activity, yielding some p-values of 10 to the minus 179, and I believe the effect size was 30% or so. So metabolic phenotyping, just kind of summarize what we've done here. We have basic system biology research. We can apply it to disease-related biomarker discovery. We can use it in health and pharmaceuticals, nutrition and lifestyle research. There are papers that look at exercise, for example, exposure to oxygen, bioprocess optimization. If you think about it, we can now help select cell lines and then optimize protein production, which equates to profits for these pharma companies. Again, indications in particular neurology, there are several papers out, and all of these papers are available on our online, and I can lead you to that website. And if you look at the various type of studies that were conducted using our platform technology to help answer some questions or develop a hypothesis. The rationale for BIOCRATES enabled system diagnostics. So I alluded to this earlier. We think in terms of phenotype. That's the thing that we can really relate to. So we can put our hands on the phenotype, and then we can start to think about how we can relate that to understanding disease, and then kind of move forward towards personalized medicine or metabolic endpoints. And these metabolic endpoints, we tend to quantify those in terms of looking at pathways, and some of the pathways we're familiar with, amino acid, energy metabolism, biogenic amines, and so forth. And then it typically translates into a signature. A disease, a phenotype has a particular signature, as I alluded to earlier in the case of the HIV and the breast cancer story there. And then we can use this now for helping develop personalized medicine, early diagnosis, some of the things we want to detect early before we can see the phenotype, the disease staging, treatment efficacy, and these other values. So a comprehensive individual system analysis. So typically we have these inborn characteristics or genomic constituency, and then we tend to think about how does that interact with the environment. We have some predisposition here, for example, HLA system, blood group, gender, really can't change that in a way. And then your acquired metabolic homeostasis. You know, there are some people that can, eat 10 hamburgers and not gain anything, and some people eat one hamburger and blows up. And then we have these somatic changes. We talked about earlier, there was some melanoma case, for example, that affects alterations. And then phenotype reveals the pathophysiology mechanism and disease-specific markers. So we look at various indications, and we understand now that there's some underlying biology pathway that's being perturbed, DNA, for example, in melanoma. And our assays now are able to then kind of scrutinize some of these pathways, whether it's mitochondrial function, whether it's oxidative stress, insulin resistant, neurotransmitters, looking at, for example, biogenic amines, and cell signaling. We talked about cell signaling earlier on, one of the speakers, and their downstream effects, upstream effects from those. And then I'll just kind of jump into the technology and products briefly. So this is a P180 kit. That was some data was showed from that earlier on, and I talked about that in terms of the same assay was used for the Alzheimer's study in Nature Genetics, Nature Medicine. And from that, we were able to look at acylcarnitines, lysophosphatidylcholines, biogenic amines, hexose. So there's 182 metabolites covering typically about 200 pathways. And if you can see where some of these particular analytes are being involved with, for example, looking at the at this particular compound over here, or serotonin in the neurotransmitter assays, the biogenic amines. So the evolution of metabolomics research. So what this is showing you now is that in earlier stages you need, lots of metabolites, and then as you develop a kind of a diagnostic approach, there's a set of metabolites that you then kind of lock in to what you're going to use, and then the number of samples. You start off with small samples, hundreds, less than 100 if you're looking at cell samples, for example. And then when you want to go ahead and validate that in the diagnostics, then you need larger sample sets. And so we fit right in here. So we are really right next to go into the diagnostics by using these quantitative approaches. And I showed you some of those relevance with those ROC curves. And the other thing about the kits and the assays is they are both validated by the European group and the FDA here in the US, and they are validated across the various platforms. You've seen some of the vendors out here, AB Science, Thermo, and Waters. So what that means is that you can conduct experiments in your various labs wherever they are situated around the world, and your data should come back very similar. And this has been one of the challenges for FDA to embrace metabolomics work, because they have typically looked at single markers. Now you can look at several markers now, which when you look at these pathways, like I alluded to earlier on in the GWAS study, where you had the two lipids, and they were surrogates for function for the enzyme. So you're looking at more than one marker now. And this is a list of the various assays we provide. So when I refer to a kit, it's actually an assay, but you can run these kits in-house, and we can run these at our facilities for you as well if you don't have the mass spec capabilities. And so the P150 and the P180, the difference between the two is that the P180 has the biogenic amines and the additional amino acids, and it also involves flow injection and an LC method as well. There's also a steroid kit that we provide as well, and that kit is currently not available in the US. However, we can run those assays, simple shipping samples out to Innsbruck. And here are a number of other assays that we provide. Neurotransmitter, there's a bile acid assay. Actually, there's a kit that's being developed for this as well that should be available here sometime mid-year. And there's a number of other assays that's available that we can run in-house for you. And these are an example of the matrices and species that we've worked with. You know, we can think about a variety of tissues, feces. I've done some study with feces. Just flash freeze, shipped to BIOCRATES. There's a slight little charge extra for us to process it out for you. The cells, you can just ship your cells, flash freeze. And Vladimir also alluded to these study design. So one of the important things in metabolomics study is to be consistent in your sample collection process protocols, so you kind of minimize human error. And if you look at the various tissues and the various species that we're able to work with. So what I wanted to talk about here is the translational research. The NIH has been pushing forward on grants. I believe Duke, MD Anderson, Stanford received some significant monies recently, several, I think $50 million each for translational research. So what I'm showing you here is that using our technology, you can look at now these longitudinal studies across the mice. And because you only need 10 microliters, you don't have to sacrifice the mice anymore. You can keep the mice going and look at how drug effects across these particular. And again, it's translational because the underlying biochemistry is very similar in cells, small animals, and humans. And just again, alluding to the kit assay, 186 metabolites, 82 samples per the 92 well plates. And here's just a time frame of doing the processing in the lab, 24 hours, 30 hours in total for the 96 tests. So in a week, you can run 500 samples in your lab. This is just a layout showing you the controls and the calibration for the LC part and the technical validation that's built into our kits, and they're highly reproducible. Again, the P180, and then I mentioned the steroid kit and what they're used for here typically. And this is our standardization of data format. You know, it's across the different platforms I alluded to earlier on, and there's the software that comes with the kit to help you understand the data. Here's a data flow where we can actually link the data out to the public domain. We can tack into the KEGG, identify what metabolites is in the KEGG database, then pull out from PubMed what literature is out there that's associated with that particular metabolite, and start developing the biology. And here is kind of a full integrated center for competency, right? So we have the know-how, we have the sample banking. You can ship samples to us, hundreds, thousands of samples, right? We have the assay portfolio. We develop custom assay as well. The kit development, we can help you with that as well, and the product on the market. I've alluded to these earlier. And I just wanted to end here by this particular doctor that our CEO bumped into in Nigeria. And he goes, is this voodoo medicine, or is it the next generation technology and system diagnosis? With that, take questions.
Thank you for the talk. I missed the point. Do you have the column come with the kit as well?
It depends on which platform you have. If it's AB-Cix, Waters, or Thermo.
Okay, so this is the one injection to get 180 metabolite, or
Yep.